Welcome to the PubMed Daily Review for April 02 ST. Today we scanned 7,039 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. Structure-guided engineering of a Cas12 nuclease unlocks Near-PAMless genome editing by Qitong Shun and Jiankong Zhu from Institute of Advanced Biotechnology, Institute of Homeostatic Medicine, and School of Medicine, Southern University of Science and Technology, Shenzhen, China. Let's break down its key contributions. CRISPR gene editing has revolutionized biology, but a major hurdle remains: the requirement for specific DNA sequences called PAMs next to the target site. This significantly restricts where we can make edits in the genome. Imagine wanting to fix a typo, but only being able to do so if the typo is next to a specific word. This paper explores a way to overcome this limitation by engineering a Cas12 nuclease. To tackle the PAM limitation, the researchers focused on a compact Cas12 nuclease. They used advanced computational models from AlphaFold to pinpoint key areas of the protein that interact with the PAM sequence. By systematically changing 38 different amino acids in this region, they aim to create new versions of the nuclease with broader targeting capabilities. These engineered nucleases were then tested for their editing efficiency and specificity. The results were impressive. The engineered Cas12 variants, particularly IKRR and STKRR, dramatically relaxed PAM specificity, enabling editing at a much wider range of sites. This expanded the targetable portion of a genome to over 25%, a fourfold increase compared to the original nuclease. Importantly, even with this enhanced on-target activity, comprehensive analyses showed no increase in off-target edits. Adenine-based editors built with these variants also achieved high editing efficiencies at endogenous sites. This study represents a significant leap forward in genome editing technology. By engineering Cas12 nucleases, the researchers have created tools that can target over 25% of the human genome, a fourfold increase. This opens doors for applications that were previously impossible due to PAM constraints. The development of these Near-PAMless editors is a crucial step towards more versatile and effective gene therapies and research. Up next, our second paper out of 5, Lenacapavir as pre-exposure prophylaxis for HIV prevention by Samaya Ibrahim and Karen Cohen from Health Systems Research Unit South African Medical Research Council, Cape Town, South Africa. Let's break down its key contributions. HIV remains a significant global health challenge, with millions of new infections each year. While pre-exposure prophylaxis, or PrEP, is highly effective, its uptake is limited, partly due to the need for daily dosing. This highlights an urgent need for more user-friendly options, like long-acting injectables, to improve prevention efforts. This study systematically reviewed 2 randomized controlled trials involving over 8,600 participants. They compared the long-acting injectable Lenacapavir, administered every 6 months, against daily oral PrEP. The researchers focused on key outcomes like new HIV infections, safety events, and mortality over a 52-week period. The results were striking. Lenacapavir led to a substantial reduction in new HIV infections, with a relative risk of just 0.07. While serious adverse events were slightly reduced, there was a notable increase in injection site reactions. Importantly, there was little to no difference in overall adverse events or all-cause mortality between the groups. This review suggests Lenacapavir is a promising new tool for HIV prevention, offering a highly effective long-acting alternative to daily pills. While injection site reactions are more common, the significant reduction in HIV infections is a major clinical benefit. Future studies should explore its long-term safety, potential for resistance, and effectiveness across a broader range of individuals. Up next, our third paper out of 5. DNA delivered by lipid nanoparticles induces CD8 T cell-dependent antitumor responses and enhances anti-PD-L1 therapy by Xiaoyan Yum and Pedro J. Sejas from Discovery Group, Spark Therapeutics Incorporated, Philadelphia, Pennsylvania, USA. Let's break down its key contributions. Immune checkpoint inhibitors have revolutionized cancer treatment, but many patients don't respond. A key reason is insufficient pre-existing anti-tumor immunity. This study explores a novel approach, using DNA delivered by lipid nanoparticles, or DNLNPs, to activate the immune system and potentially overcome these limitations. To investigate this, researchers used various animal cancer models. They delivered non-coding DNA encapsulated in lipid nanoparticles directly into cells. The study focused on observing the immune response, specifically CD8 plus T cell activity and the antitumor effects, both alone and in combination with anti-PD-L1 therapy. The results were compelling. DNA-LNP treatment led to a robust recruitment of CD8 plus T cells to the tumor site. This immune activation resulted in potent CD8 plus T cell-dependent anti-tumor effects and significantly improved survival across different cancer models. Importantly, DNA-LNPs showed remarkable synergy with anti-PD-L1 therapy, extending survival even in tumors resistant to ICI treatment. This study highlights DNA-LNPs as a promising new immunotherapy approach. By activating the immune system, they can enhance anti-tumor responses and potentially overcome resistance to current therapies like anti-PD-L1. These findings pave the way for future clinical investigations into DNA-LNPs, either alone or in combination, to improve cancer treatment outcomes. Up next, our fourth paper out of 5. First human cell-based cultivation system for the syphilis spirochete treponema pallidum by Uri Bozak and David Smajas from Biology, Faculty of Medicine, Masaryk University, Brno, Czech Republic. Let's break down its key contributions. For over a century, studying the syphilis-causing bacterium, treponema pallidum, has been a challenge. Traditionally, researchers have relied on infecting laboratory rabbits to grow the spirochete. While a breakthrough in 2018 allowed cultivation in rabbit cells, a truly humanized system is still needed. This is essential for unlocking deeper insights into how this persistent pathogen interacts with its human host and causes disease. To address this, the researchers tested 3 human foreskin fibroblast cell lines, comparing their ability to support the growth of treponema pallidum against the established rabbit epithelial cell system. They used 8 different strains of the spirochete, monitoring their growth over a 10-week period. The key was to see if these human cells could effectively cultivate the pathogen, mimicking a more natural host environment. The results were promising. All 3 human cell lines supported the growth of T. pallidum, with one strain, DEL1, being continuously cultivated on human fibroblasts for over a year. While the overall growth rate was a bit slower compared to rabbit cells, the relative replication rates between different strains remained consistent with previous findings. This represents a significant step towards a more humanized in vitro system. So, what does this mean for syphilis research? This study demonstrates that human cells can indeed be used to grow treponema pallidum in the lab, moving us closer to understanding this disease in a human context. This breakthrough opens exciting new avenues for investigating how the spirochete causes infection and how our bodies fight back. Ultimately, this could lead to better diagnostic tools and more effective treatments for syphilis. Up next, our 5th paper out of 5. Single-cell multi-omic human brain atlas reveals regulatory drivers of cortical regionality by Carter R. Palmer and Kuan Zhang from Neurological Diseases, Sanford Burnham Prebys Medical Discovery Institute, La Jolla, CA, USA. Let's break down its key contributions. The human brain is incredibly complex, with different regions specialized for different tasks. But how do these specialized cells and regions arise? We don't fully understand the molecular instructions that guide this process. This study aims to shed light on the gene regulatory networks that dictate cell identity and location within the human cortex. To tackle this, researchers analyzed over 3 million cells from 9 different regions of the human brain. They used advanced multi-omic techniques to look at gene expression, chromatin accessibility, and spatial location all at once. This allowed them to build a detailed map and identify the key players, like transcription factors, that control how cells become specialized and where they end up. The analysis revealed fascinating patterns. They found a clear gradient in calcium regulatory machinery from the front to the back of the brain. They also observed specific changes in signaling receptors along the sensory to transmodal axis. Crucially, they pinpointed gene regulatory networks and transcription factors that seem to be responsible for these cell type and region-specific differences. This work gives us an unprecedented look into the molecular organization of a human brain. By understanding these fundamental regulatory mechanisms, we can better investigate how things go wrong in brain development and disease. It's a significant step forward in deciphering the complexity of a human cortex.